Phylogeny and Cladistics. Phylogeny. Phylogeny is the evolutionary history of a species or group of species. The sequence of the species or group that have evolved from a common ancestor is known as a lineage. A clade is a group of organisms that share a common ancestor and includes all the lineal descendants of that ancestor. Phylogeny can be studied using phylogenetic trees and cladograms. Both methods represent hypotheses about evolutionary relationships between lineages and are constantly being revised based on evidence. Phylogenetic trees. A phylogenetic tree is a branching diagram illustrating phylogeny, the evolutionary relationships between species. Phylogenetic trees also show time frames of the different species' existence. This is an example of a mammalian phylogenetic tree. Phylogenetic trees. Phylogenetic trees are used to show relatedness among organisms. Branches separate organisms based on traits they have in common. Biologists use two methods to place organisms on the phylogenetic tree: molecular similarities. Similarities in DNA, RNA, and proteins. These provide the most accurate and reliable placings of the organisms on the trees. Morphological similarities. Similarities in body structure and embryonic development of living and fossil species. Phylogenetic trees were originally entirely based on morphology, but as our knowledge of DNA and genetics grew, many changes were made to the trees. Phylogenetic trees are based on research and hypotheses, and are constantly updated with new evidence. How to read a phylogenetic tree. The diagram shows a clade. Start with the root, the common ancestor at the bottom or left of the diagram. Follow changes in biodiversity over the passage of time by moving upward or rightward. So the last common ancestor of all species that appear later are further up on the diagram. But the root, the last common ancestor of the clade, is at the bottom of the diagram. How to read a phylogenetic tree. Each branch point, or node, locates the common ancestor of all taxa that follow. A speciation event in that common ancestor led to the divergent branches that follow. Cladistics studies descent with modification. One of the tools used to create a phylogeny is cladistic analysis. A cladogram is a special type of phylogenetic tree that uses shared and derived characters to determine which species are most closely related. It may consider traits both lost and gained in evolution. Shared characters are traits that are present in two or more lineages, branches on the tree. Derived characters are traits that occurred in the most recent common ancestor and were passed on to all the descendants. Unlike a phylogenetic tree, no times are shown on a cladogram. Analyzing a cladogram. The evolutionary relationships between a group of mammals is shown below. The outgroup is the turtle, a distantly related species that serves as a basis for comparison. The shared ancestral character that groups these species together is hair, a trait of all mammals. The other traits in red are derived characters, features unique to a branch that were developed in evolution. Another version of the cladogram. The common ancestor of reptiles and vertebrates is shown at the root before the hair. Domestic cats have all derived characters studied, so this cladogram tells us about their evolutionary history. In this version of the cladogram, the common ancestor of all animals is shown at the root, and hair is the shared character that defines this group of species. Another version of the cladogram. Carnivory is a derived character that separates the horse lineage from wolves, leopards, and domestic cats. The most recent derived character shown is the ability to purr, and it separates domestic cats from other species. Clade. In this cladogram, clades are shown in different colors. When species share more characteristics, then they share more evolutionary history, and they have a closer evolutionary relationship. Character tables. A character table allows us to record characters and reveal evolutionary relationships. It can be used to build a phylogenetic tree. This character table was used to build the cladograms on the previous slides. 0 indicates the character is absent, and 1 indicates the character is present. The more derived characters shared between two species, the more closely related they are.